Hi, I'm Sue Biggins, and I'm an investigator at the Fred Hutchinson Cancer Research Center in the Division of Basic Sciences. I'm also an investigator with the Howard Hughes Medical Institute. Today, I'm going to talk about the process of chromosome segregation, which ensures that your cells inherit the right chromosomes when they divide. Cells must inherit the right chromosomes. This is a karyotype of a normal cell. These are chromosome spreads, and you can see that you have two copies of every chromosome, one from mom and one from dad. This looks really different in a cancer cell. Here is a cancer cell chromosome spread, and you can see a really large variation in chromosome number. There's entire gains and losses of chromosomes. In addition, there's chromosomal rearrangements that often occur. This condition where cells have the wrong number of chromosomes is called aneuploidy, and it's one of the most common hallmarks of cancer cells. And you can learn a lot more about the consequences of aneuploidy in the lectures from Angelica Amon. To ensure that daughter cells inherit the right chromosomes, they undergo two major events during the cell cycle. After G1, or interphase, they enter S phase, and the chromosomes replicate or duplicate to form sister chromatids. At mitosis, the chromosomes segregate to opposite poles, and then when the cells undergo cytokinesis to form daughter cells, this ensures that each daughter cell inherits one copy of each chromosome. Now, a chromosome missegregation event during mitosis, shown here, results in aneuploidy, as I just showed you on the previous slide, which is a common hallmark of cancer. And I should mention it's a common hallmark of other diseases such as birth defects. And I want to remind you that you have greater than 1 trillion cells in your body. So this process has to happen correctly for every pair of chromosomes in every cell every time they divide. So mitosis was first observed in the late 1800s by Walter Fleming. And these are just drawings he did. What he did is he stained grasshopper cells, or salamander cells, with a DNA dye, and then he was able to deduce that during the cell cycle, chromosomes duplicate and partition. And you can see here that they partition. He even drew in fibers that we now know are microtubules. Due to major advances in cell biology and microscopy, we can actually visualize this process in real time. I'm going to show you two movies from Herrick Kops's labs where he visualizes mitosis. What he did, or what their lab did, was they expressed a fluorescent fusion to a histone so that they could visualize the chromosomes, and then they marked the microtubules with a fluorescently tagged component of the microtubules. So I'm going to start the movie, and what you're watching here are the microtubules forming a spindle, and those microtubules are capturing the chromosomes, aligning them, and they're pulling them to opposite poles. Now, this is a really dynamic and complicated process, and it turns out it's actually also very error-prone. I'm going to show you an example of that in the next movie. Here, most of the chromosomes get properly attached to microtubules, but in the upper left, you can see an entire set of chromosomes doesn't get captured, and therefore it doesn't segregate to opposite poles, and that generates the aneuploidy that I told you is a hallmark of many diseases. So let's break down the events of chromosome segregation. As I said, during S phase, chromosomes replicate to make sister chromatids, shown here, and then a complex called cohesin physically links those sister chromatids together. And that's important so that the cell can distinguish pairs of chromosomes. At metaphase, a bipolar spindle forms, and this is due to the microtubules that emanate from the spindle poles, and some of those capture a locus on the chromosome called the kinetochore. And we're going to talk about the kinetochore in depth today. Once every pair of chromosomes is properly captured by microtubules, a complex called the APC, or anaphase promoting complex, is activated. And it cleaves cohesin so that the linkage is destroyed, and that allows the microtubules to physically pull those chromosomes to opposite poles at anaphase. Now, the kinetochore, as I said, is the structure that directs segregation. This is a really enormous protein complex that assembles on centromeric DNA. And so this is just a cartoon here that you can see the kinetochore. The centromere I'm going to define as the DNA on the chromosome where the kinetochore forms. And in this image, you can see that the inner centromere is the underlying DNA. And then there's an inner kinetochore, which are the proteins that directly bind to the centromeric DNA. And then there's a whole nother layer of proteins that assemble onto that, and they form the outer kinetochore. And that is the microtubule attachment site. And we now know that the human kinetochore has more than 100 unique components. So it's a really complicated structure, and most of these proteins are present in multiple copies. So there's hundreds and hundreds of proteins in just a single kinetochore, one of the most complicated protein structures in the cell. The centromere is defined as the chromosomal site of kinetochore assembly, as I said, and you can see it's the primary constriction in these images that you can see here. When we look at this by electron microscopy, the underlying chromatin is this sort of fibrous mat underneath, and then you can see the structure on top of that, where the white arrows are, is the kinetochore. And then you can actually see the microtubules are attaching to that kinetochore. And I think you can appreciate that it's very difficult to really understand the structure of this complex protein structure from looking at EM images in cells. There's 3 types of centromeres: a point centromere, a regional, and a holocentric. And they're defined by the number of microtubules that the kinetochore can bind. So point centromeres bind to just a single microtubule. Regional kinetochores are the most common, and they consist of multiple microtubule binding sites in a single kinetochore on the centromere. And then holocentric chromosomes are really interesting. They're microtubule attachment sites over the entire chromosome, and that's common in insects and worms. Now, centromeres vary a lot in size and sequence as well, and this is just representative centromeres from a number of organisms to make this point. So in budding yeast at the top, there's 125 base pair centromere. Just 125 base pairs is sufficient to ensure that that's a centromere. And this is really different in other organisms, such as humans and Arabidopsis, that have megabases of DNA that form the centromere. They also vary a lot in sequence. Budding yeast is exceptional, and it has a sequence-specific centromere that's sufficient to drive kinetochore formation. However, in most organisms, such as the rest of them shown here, the hallmark of the centromere is really just repetitive DNA. And so in humans, it's just repeats of alpha satellite arrays. So the underlying sequence is highly repetitive, and it's usually AT-rich. And because of this, the last region of the genome is usually the centromere to be sequenced because it's such highly repetitive DNA. Now, because there's really no sequence specificity to centromeres other than budding yeast, it turns out that centromeres are normally epigenetically defined or specified. So the site on the chromosome that's going to become the kinetochore is defined epigenetically, and the most common hallmark is this nucleosome called CEMP-A. So as you know, the chromosome consists of chromatin, which is built up of nucleosomes. Normally, those nucleosomes contain H2A, H2B, H3, and H4. However, at the centromere, there's an interesting nucleosome called CEMP-A, where the 2 copies of H3 are replaced with this histone variant called CEMP-A. And so what this means is at the centromere, there's arrays of CEMP-A nucleosomes interspersed with H3 nucleosomes. And this histone variant is only present at centromeres. So these nucleosomes are exclusively at the centromere and are clearly the epigenetic mark. Another hallmark of centromeres is that they're embedded in heterochromatin. In addition, there are specific modifications to the histones in these centromeres, so they have a lot of unique features. And the field is still trying to understand how this histone variant is deposited specifically at the centromere and how that's controlled, and then what are the mechanisms that make sure that it doesn't accidentally go into the rest of the chromosome where it could form additional kinetochores. Okay, so let's turn to the kinetochore now. The first kinetochore proteins were identified by exploiting the fact that it turned out that patients who have some autoimmune diseases have antibodies that recognize the centromere or kinetochore locus. And you can see that in this image from Bill Earnshaw. In the top image of a cell, immunofluorescence image, you can see tubulin shown in blue. And at the tip of those kinetochore-microtubules, you can see the red dots that are the antibodies from these patients. And that's called ACA, anti-centromere antibodies. So Earnshaw and independently another group, Goldner, realized that they could take advantage of the fact that these antibodies recognize kinetochores to clone the first kinetochore proteins. And actually in this image below, you can see CEMP-A staining. It turned out that CEMP-A that I just talked about was the first kinetochore protein that Earnshaw identified. Over the years, biochemistry and genetics have really identified the bulk of kinetochore proteins. And these are immunoprecipitations from Ian Cheeseman, where you don't need to worry about what all these complexes are or what all these proteins are. The point I'm trying to make is one of the main approaches to identifying kinetochore proteins was to take one kinetochore protein, immunoprecipitate it, and to identify its co-purifying proteins. And over the years, we learned that one of the hallmarks of the kinetochore is that it can be broken down into subcomplexes, stable subcomplexes that can be purified on their own and reconstituted on their own. In addition, I should comment that genetics have, of course, contributed heavily to identifying these proteins as well. Model organisms such as budding yeast, fission yeast, Drosophila worms, people did genetic screens to look for things like chromosome loss, mutants, and those screens additionally identified many kinetochore proteins. So what we know now is that there are many subcomplexes, and I won't talk about all of them. And the basic idea is that they form a hierarchical kinetochore structure, and that's just cartooned here. There are many, many copies of these subcomplexes, and so in this cartoon, this is greatly simplified. But the things to remember are that, as I said, there's a centromeric chromatin structure at the bottom that's the foundation for the kinetochore, and a hallmark is the Sempe nucleosome. The inner kinetochore forms on top of that, and this is often called the CCAN for constitutive centromere-associated network. These are proteins that then bind to that centromeric chromatin and form the foundation of the kinetochore. They're present throughout the cell cycle. And then at mitosis, a number of outer kinetochore proteins assemble onto that base. And as I said, there's many subcomplexes, some of which directly bind to the microtubule. Okay, so now that I've told you about the structure of the kinetochore, let's talk about its functions. And there's three major functions that kinetochores carry out. One is a mechanical role, where they maintain an attachment to the tip of the dynamic microtubule. A second is that they have interesting biophysical properties. Tension can stabilize these attachments, and we'll talk about that. And finally, they serve as signaling hubs. Kinetochores can completely halt the cell cycle if there's a defect in kinetochore-microtubule attachments. And so I'm going to talk about each of these functions. Let's start with the mechanical role. So to remind you, microtubules are the polymers that segregate the chromosomes, and these are hollow protofilaments that are assembled from alpha and beta tubulin dimers. And they exist in one of the four states shown here. They're either assembling due to the addition of tubulin subunits at the tip, or they're disassembling due to their removal. And when they switch from assembling to disassembling, it's called a catastrophe event. And when they switch from disassembling back to assembling, it's called a rescue event. And this process where microtubules in a population are constantly growing and shrinking is termed dynamic instability and was originally discovered by Hutchinson and Kirshner. Okay, so the kinetochore has a hard job. It has to stay attached to these microtubule tips. Kinetochores have to make load-bearing attachments. Now, the microtubules have an inherent polarity. The minus end is embedded in the poles, and the plus end is defined as the distal end. So remember, microtubules are growing and shrinking in the population, and at some point, a microtubule will capture a kinetochore. And most of the time, this is a lateral attachment where the kinetochore initially is on the side of the microtubule. Now, if things stayed like this would not result in proper chromosome segregation. Instead, it has to get converted to a tip attachment, shown here, where the kinetochore is attached to microtubules from opposite poles. And remember, I told you, thousands of tubulin subunits are being added and lost from this tip while that kinetochore has to stay bound. So it's almost like you're trying to climb a rope and someone's constantly pulling the rope out from under you. So the kinetochore has to stay bound to this really dynamic tip. And so a huge question is, how can that happen? So over the years, we've identified proteins within the kinetochore that bind to the microtubule, and I'm only going to talk about one of those today, which is NDC80, because it's really considered to be the major microtubule binding activity within the kinetochore. This is the structure of a domain of the NDC80 protein itself, which was originally solved by the Harrison lab, and you can see it's a CH domain, which stands for chalconin homology, and this is commonly found in other microtubule binding proteins such as EB1. Now, Andrea Musacchio's lab solved an engineered structure. NDC80 is a 4-protein complex. They made this engineered complex called the bonsai and discovered that there's actually 2 chalconin homology domains, one in NDC80 and one in UF2. And then over the years, we've realized that NDC80 has the major microtubule binding activity. This is higher structural information from Eva Nogales' lab. The microtubule is shown in green. And you can see NDC80 is shown in blue, and you can see the calponent homology domain directly binding to that microtubule. The NUF2 is shown in yellow, and you can see that it doesn't directly bind. And I want to point out the red is another part of NDC80, which is an N-terminal extension that also has microtubule binding activity. So this is the complex that really is considered to be the major microtubule binding activity. However, I want to emphasize there really are other microtubule binding proteins in the kinetochore, and we're still trying to understand how they all act together and what their relative contributions are. So now let's talk about the mechanism that the kinetochore can use to actually maintain an attachment to these dynamic tips. So there are two major models right now. The first is the conformational wave model, and the idea here is that the microtubule stores a lot of energy, and when it switches from assembling into disassembling, the tip structure peels back, the protofilament curves back, and as long as the kinetochore can maintain an attachment to this curved protofilament, it can harness the disassembling energy of the microtubule to move the chromosome. And there's two ways that you can imagine that the chromosome can stay attached. One is a ring model where a part of the kinetochore forms a ring around the microtubule, and there's evidence in budding yeast that the DAM1 complex does form a ring. There's also a fibril-based mechanism where individual components of the kinetochore can attach to the microtubule tip and then maintain an attachment through this conformational wave. Now, Hill proposed a different model called bias diffusion. In this model, the idea is that the kinetochore contains multiple weak microtubule binding elements that rapidly bind and unbind to the microtubule tip. And as long as this diffusion is biased towards the tip, which can occur due to thermal fluctuations of the chromosome binding near the tip, that then the kinetochore would stay attached to the growing and the shrinking tip. And there's evidence that this model might be true because the Asbury and Davis labs showed that the NDC80 complex itself can undergo bias diffusion. Finally, there's hybrid models that would take both of these ideas into account. And so we're still actively trying to understand what the underlying mechanism for attachment really is. Okay, so now let's talk about the interesting biophysical properties of the kinetochore where tension can stabilize rather than destabilize an attachment. Kinetochores have to make bioriented attachments, so each sister kinetochore has to bind to microtubules from opposite poles. However, as I said at the beginning, the process of making attachments is actually very error-prone. And there's a number of incorrect attachments that can be made, such as mono-oriented attachments where one or both sister kinetochores attach to microtubules from a single pole. There's even mirrotelics shown here on the bottom, where one kinetochore can attach to microtubules from both poles. And it's essential that the cell can detect and correct these errors. And so how can cells detect and correct these errors? It turns out that tension is the key. Now, Dietz originally noticed that chromosomes would continually reorient on the spindle until they became stably bioriented. And then Bruce Nicholas really did the pioneering experiment to show that that's because tension is stabilizing the attachments. So what he did is take advantage of the fact that grasshopper spindles are really large and that you can use micromanipulation techniques. So he used meiosis I spindles where now the homologs are attached to opposite poles rather than the sister chromatids. And he took a micromanipulator and was able to convert a bi-oriented attachment where the homologs are attached to opposite poles into one where the homologs are attached to the same pole. And he noticed that this is highly unstable, and what would happen then is that the kinetochore would detach. And there were one of two fates. If it went back and created the wrong attachment again, that was unstable. However, if it created the correct attachment shown in the top, it was now stabilized. And then to demonstrate that this is because of tension, he actually used a needle to artificially apply an opposing force. So he took that incorrect attachment, where both homologs are attached to the same pole, applied an opposing force, and now the incorrect attachment was stable. And this really demonstrated that that's because tension is stabilizing that attachment. So tension stabilizes attachments, and they're in vivo very stable once they come under tension. And in contrast, any attachment that's lacking tension is highly unstable. So how can tension stabilize attachments? And we know that there's at least two mechanisms that contribute to this. There's work from a number of labs that have shown there's an error correction pathway that uses phosphorylation by the Aurora B protein kinase. So the idea here is that when there's an incorrect attachment that lacks tension, the Aurora B kinase recognizes that and can phosphorylate kinetochore proteins. And I want to mention that NDC80 is one of the major targets of the Aurora B kinase. When these proteins are phosphorylated, it destabilizes that incorrect attachment, and then that gives the cell a chance to go back and make the correct attachment again. Now there's a second mechanism, and that's one where tension directly stabilizes attachments. And the idea here is that tension itself promotes more microtubule binding elements to bind to the tip of the microtubule. And this is like those finger traps, if you remember, where as you pull on them, things get even tighter. And this is really just an intrinsic property of the kinetochore-microtubule interaction. There is no Aurora B involved in this. And in part 2 of my talk, I'll tell you how we discovered this mechanism. Now let's turn to the signaling functions of the kinetochore. And as I said, the kinetochore can transmit a signal to completely halt the cell cycle. Cell cycle progression has to be halted when kinetochore-microtubule interactions are incorrect. So when there's a lack of tension or an unattached kinetochore, the checkpoint has to recognize that and halt the cell cycle. And the spindle assembly checkpoint, I'm going to call it the spindle checkpoint for short, is the signal transduction system that can do that. Genetic screens from Andy Hoyt and Andrew Murray's labs originally identified the checkpoint genes. And the idea here is they use budding yeast as a model organism. As you know, the yeast will divide and make a colony when they're grown under normal conditions. If you add a microtubule depolymerizing drug such as Benomyl, this creates difficulty for the cell in making kinetochore-microtubule attachments because now the microtubules are less stable. And then there's a mitotic delay while the cell tries to make the proper attachments, and that's due to the spindle checkpoint. So the Hoyt and the Murray labs reasoned that mutants in the checkpoint would not be able to delay, would therefore go through the cell cycle with incorrect attachments, and that would lead to cell death. So MAD2 is one of the checkpoint mutants, and when they treated that with Benomyl, there was no delay, and then they just get a microcolony of dead cells. So by looking for mutants that cannot grow on Benomyl, they were able to isolate really the majority of spindle checkpoint genes. We now know those spindle checkpoint genes that were originally identified in budding yeast are conserved. And there are additional ones that they didn't identify, but the bulk really were identified in these 2 classic screens in the early 90s. Okay, so where is the checkpoint signal generated? The first homolog of the yeast genes identified was the Xenopus MAD2 gene, and the Murray lab showed that it's required for the checkpoint using frog egg extracts. And then they made the striking finding that MAD2 binds specifically to unattached kinetochores. So you can see that in this image here from their paper. This is a newt lung cell, and this was a collaboration with Ted Salmon's lab. And you can see on the left the chromosomes, that most of them have aligned at the metaphase plate and are properly attached to microtubules. And then on the right is immunofluorescence against MAD2, and you can see there's almost no MAD2 staining. So the kinetochores that are properly attached do not have any MAD2. But if you look on the right-hand side, or on the left-hand side, you'll see a mono-oriented chromosome. One kinetochore is attached to the pole and the other one is not. And then if you look at the staining of MAD2, you can see it specifically stains the unattached kinetochore in that pair of chromosomes. So this really was the evidence that MAD2 specifically binds to unattached kinetochores, and over the years, a lot of work has shown that kinetochores clearly generate the checkpoint signal. And we now know there's a phosphorylation cascade that recruits checkpoint proteins to unattached kinetochores to trigger the checkpoint, and I'm not going to talk about all those details. Now, what is the checkpoint monitor? I've already told you that defects in tension or attachment will lead to aneuploidy and chromosome segregation. So it's critical that both of these defects are monitored. And it's been really controversial whether tension or attachment is the key to triggering the checkpoint. Work from Conley Reeder suggested that attachment is monitored. So he did a classic experiment where there was a cell that had a mono-oriented unattached chromosome. So there's a single kinetochore attached to the microtubules and the other is unattached. He went, and those cells are arrested because the checkpoint is on because there's a defect. Now, he went in and he laser ablated that unattached kinetochore. And that satisfied the checkpoint and the cell cycle continued, even though the remaining kinetochore is attached and not under tension. And this was the work that really suggested that unattached kinetochores were what the checkpoint was monitoring. However, remember that classic experiment I told you from Bruce Nicholas, where there was a mono-oriented chromosome attached, where both sister kinetochores are attached to the same pole. And in that situation where there's no tension, but there is attachment, it turns out that the cell cycle was stopped due to the checkpoint. And when he went in and micromanipulated and applied the opposing tension, it turned out that the cell cycle went ahead, even though the kinetochores were attached and to the wrong pole. And so this showed that tension seemed to be sufficient to tell the cell that everything was ready to go, even though those attachments were incorrect. So it's still really confusing whether tension or attachment is the primary signal. And I just want to remind you that defects in tension are converted to unattached kinetochores. So remember, when kinetochores lack tension, these attachments are released due to error correction mechanisms, and that generates unattached kinetochores. So the reason it's been so hard to understand might just be because these two issues are so interrelated. Now let's talk about how the checkpoint halts the cell cycle. A clue to this came from work from Ted Salmon's lab that looked at the MAD2 protein and found that it dynamically cycles on and off the kinetochore. So what they did here is they took cells expressing a MAD2 GFP fusion so that the kinetochores, they could see the kinetochore, MAD2 on the kinetochores, and they treated the cells with nicotazole to generate lots of unattached kinetochores, which is why all of these kinetochores are lighting up with MAD2. They then did what we call a photobleaching experiment where they ablated in a single kinetochore shown there, the MAD2 fluorescence, and then they watched how long does it take for new MAD2 to bind to the kinetochore. And what they learned from this experiment is that a population of MAD2 rebinds within 25 seconds, which is extremely rapid. And I want to point out there was also a more stably bound population as well. So this dynamic localization of MAD2 suggested that perhaps it forms an inhibitory complex when it goes to the kinetochore that can quickly diffuse and then generate a cell cycle arrest. We now know that the APC is the target of the checkpoint. I told you earlier that this anaphase promoting complex promotes the metaphase to anaphase transition. And it turns out there's an activator called CDC20 that triggers the APC activity. The spindle checkpoint directly inhibits this protein. MAD2 binds to CDC20, and additional proteins do as well, and this complex is called the MCC, or the mitotic checkpoint complex. And when that complex binds to CDC20, it prevents it from activating the APC. So Hangtao Yu and Andrea Musacchio's lab solved the structure of MAD2, and they found out there's actually two forms. There's a closed form, and that directly binds to CDC20 and inhibits it, and then there's an open form. And so the idea here in this template model that Andrea proposed is that there's a cytoplasmic pool of MAD2 that's in the open form, and then there's actually a closed form of MAD2 sitting on the kinetochore. When the open form binds to the closed form on the kinetochore, it gets converted to the closed form that then diffuses away and can bind to CDC20 to inhibit it. I just want to summarize the general model for the checkpoint right now. A lot of details are missing, but you'll get the overall picture. The idea here is that there's a MAD2 complex, and MAD1 is its receptor. That complex binds to the kinetochore, such as an unattached kinetochore. And then open MAD2 binds to the MAD2 that's closed on the kinetochore. That converts it to closed MAD2. That closed MAD2 binds to CDC20, and then that assembles into a mitotic checkpoint complex that then can go inhibit the APC. And so that keeps the cell cycle off while the proper attachments are made. Once everything's okay, that checkpoint complex is relieved, and CDC20 is then activated to then go bind to the APC and promote cell cycle progression. And so we're still working out a lot of details of this model, but that gives you a general picture of how the checkpoint is thought to work. Okay, so what I've told you about today are the complex functions of kinetochores. They play mechanical roles in directly coupling the kinetochore and chromosome to microtubules to move the chromosomes during the cell cycle. We've identified some of the major players that bind to microtubules, such as NDC80, but we're still trying to identify what other microtubule binding proteins are in the kinetochore, what are their relative contributions to maintaining an attachment, and really what is the underlying mechanism, conformational wave, bias diffusion, by which the kinetochore can maintain an attachment. Kinetochores have these really interesting biophysical properties where tension actually stabilizes attachments, and we know that there's at least two mechanisms that contribute to that, an Aurora B mechanism. We still don't know how Aurora B selectively works on kinetochores lacking tension, but doesn't affect those that have tension. And then, as I've said, we've identified a second mechanism, an intrinsic mechanism that we're still trying to understand the underlying mechanism for. And then finally, they can act as signaling hubs where a single unattached kinetochore can completely halt the cell cycle through the spindle checkpoint. And we're still trying to understand what the checkpoint is monitoring, what state of kinetochore-microtubule attachment, and then there's a lot of work to be done to understand how that signal is amplified to halt the cell cycle, and then how, when everything's set up correctly, is that checkpoint silenced so that then the cell cycle can continue. So I'm going to talk specifically in my next talk about the first two functions, and I thank you for your time.